All right, ready for today's deep dive? We're going way beyond just DNA and genes this time.
Oh, this is going to be good.
We're talking chromatin and the histone code. It's like the instruction manual for how our cells pick and choose which genes they actually use.
Yeah, it's amazing, right? Like, how can identical twins have the same DNA but end up with different traits, maybe even different health outcomes?
Exactly. So chromatin is the key here, I guess.
It is. You know how we always picture DNA as this perfect little double helix?
Yeah, neat and tidy.
Well, in reality, it's way more complicated. Think about stuffing a huge, tangled up string of lights into a tiny little box.
Oh, I see.
That's kind of what's going on in our cells. And to keep everything organized and under control, DNA is packaged up with proteins, forming this complex called chromatin.
So DNA isn't just like floating around in there.
No way. It's all about organization. In the nucleus, DNA wraps around these proteins called histones, like thread on a spool,
Okay.
And these DNA histone units, they're called nucleosomes, the basic building blocks of chromatin.
So I'm picturing these little spools of DNA all lined up nice and neat. But how does the cell know which parts of the DNA it needs to read?
That's where the histone code steps in.
The histone code.
Yeah, think of it as a language made of chemical tags. They tell the cell what to do with each part of the DNA.
Chemical tags. So there's even more to DNA's instructions than just the A, T, C, Gs.
You got it. See, the histone proteins, they have these tails sticking out.
Okay.
And those tails can be modified, with different chemical groups.
Huh.
Acetylation and methylation, those are two of the big ones, like little flags attached to the histones, signaling whether a gene should be turned on or off.
So it's like those tags are giving extra instructions beyond the DNA code itself.
Precisely. For instance, acetylation. It's like loosening up the chromatin structure.
Loosening it up how?
Making it easier for the cell's machinery to access the DNA and read the genes in that section, like opening a book to the chapter you want to read.
Makes sense. And methylation. Is that like closing the book, keeping that chapter hidden?
It can be, but it's not always that simple. Methylation's more complex.
Oh.
It can actually activate or repress genes, depending on where it happens. More like sometimes it highlights a specific passage in the book, other times it bookmarks a page for later.
Oh, I see. So we've got these tags on histones, and they can tell a gene to turn on or off. But who's putting those tags on? And who's reading them? Sounds like a whole team of cellular workers managing this code.
You're exactly right. There are these specific enzymes, and they're responsible for writing, erasing, and reading this histone code.
Wow.
We call them writers, erasers, and readers.
Like a library.
Exactly. You've got librarians adding labels to books, that's writers. Librarians taking off old labels, that's erasers. And readers, well, they use those labels to find the info they need.
I like that. So writers are adding the tags, erasers are removing them, and readers are checking the tags to control which genes get accessed.
You got it. For example, histone acetyltransferases, we call them HATs, are writers that add acetyl groups. They're like the librarians who slap a highly recommended sticker on a book.
Okay.
On the flip side, histone deacetylases, or HDAses, are erasers. They take those highly recommended stickers off when a book, isn't so popular anymore.
So what are the readers doing with all this information?
Readers are proteins that recognize specific modifications on those histones. They recruit other proteins to activate or repress the transcription of a gene. It's like someone sees a highly recommended sticker and decides to check out that book.
Got it.
And this back and forth between the writers, erasers, and readers, that's what keeps the histone code dynamic, responsive to what the cell needs at any given time.
So it's not just a set of fixed instructions. This code's constantly being updated.
Exactly. And
Right.
chromatin structure itself, it's not fixed either. It's constantly being remodeled.
Remodel.
Like rearranging furniture in a room to make more space or change the flow.
That's a good way to think about it.
Yeah.
So who's doing the rearranging in this cellular furniture store?
We have these amazing molecular machines called ATP-dependent chromatin remodelers. They use energy to move around, even kick out or just restructure those nucleosomes we were talking about.
Wow.
Imagine them sliding nucleosomes along the DNA or even kicking them out completely. That way, other proteins have room to get to the DNA.
Okay, I think I'm following. But earlier you mentioned how identical twins can have different traits, even though they have the same DNA. Where do those differences come in?
That's a great question. It leads us to histone variants. You see, we've been talking about the core histones that make up most nucleosomes.
Right.
But there are also alternative versions of these histones, and they have very specific jobs.
Wait, so there are different types of histones? It's like finding out there's not just one kind of spool for thread, but all sorts of specialized ones.
You got it. These histone variants can become part of nucleosomes, adding even more layers to the histone card.
Sure.
For example, there's a variant called CENPA. It's super important for cell division.
CENPA. That sounds important. What does it do?
CENPA. It hangs out at the centromere. That's a part of the chromosome that's vital for chromosomes to separate properly during cell division.
So it's like a specialized histone for a specific job.
Exactly. Think of CENPA like a special spool, designed for a particular type of thread. It makes sure that thread is organized and distributed correctly when the cell divides.
Oh, wow.
And here's the kicker. Research has shown that if CENPA isn't working right, it can mess up chromosome segregation, potentially even leading to cancer.
So these variants aren't just minor players. They can have a big impact on our health.
Absolutely. We're only just beginning to figure out everything they do and how they add to the complexity of the histone code.
So we've got histones, chemical tags, writers, erasers, readers, even specialized histone variants. And you said if this system gets messed up, it can cause problems like cancer.
Yeah. That's right. But before we get into the histone code and disease connection, there's another big player we need to talk about, DNA methylation.
Hold on, there's more. We've got all these modifications on the histones, and now we're adding modifications to the DNA itself.
Yep. DNA methylation is when a methyl group is added straight to the DNA molecule, usually to a cytosine base. It's like another level of control on top of the histone code.
So we've got these methyl groups attaching to DNA. What do they do?
Think of it like a more permanent way of silencing a gene. Histone modifications can change quickly, but DNA methylation is more stable, it sticks around longer. It's like locking up a chapter in the instruction manual, so it's hard to get to.
And this DNA methylation works with the histone code to fine-tune gene regulation.
Exactly. They often work together, like areas of DNA with a lot of methylation usually have tightly packed chromatin and repressive histone modifications too. It's like multiple layers of security to make sure certain genes stay inactive.
Okay, so we've got this interplay between histone modifications and DNA methylation. But how does all this impact our health?
Well, remember how we talked about cell differentiation earlier? How one fertilized egg turns into all the different cell types in our body? The histone code and DNA methylation are essential for making that happen.
So it's not just turning genes on or off. They're guiding cells to specialize too.
Yep. Imagine a stem cell. It could become any cell type. But when it differentiates, like into a heart muscle cell, certain genes need to be turned on and others need to be turned off permanently. That's all thanks to a carefully coordinated dance of histone modifications and DNA methylation.
So these epigenetic mechanisms are creating a roadmap for each cell, guiding it to its destiny.
That's a great way to put it. And if these mechanisms go wrong, it can lead to developmental problems or even diseases like cancer.
We've talked about cancer a few times now. Can you give me an example of how the histone code going haywire can cause cancer?
Sure. Let's look at a gene called P53. It's a super important tumor suppressor gene.
So if P53 is doing its job, it stops cells from growing out of control.
Right. But if the histone code around the P53 gene is messed up, the gene can't be expressed. Imagine if the highly recommended sticker on a book about preventing cancer got ripped off and the book got lost in the library.
So even though the P53 gene is there, if the histone code is messed up, it's like the cell can't access the instructions.
Exactly. And without P53 doing its job, cells can start dividing like crazy and maybe even form a tumor.
That's scary. So these disruptions to the histone code, do they just happen randomly, or are there certain things that can trigger them?
That's a complicated question, and we're still trying to figure out all the pieces to that puzzle. Sometimes it happens because of mutations in the genes that code for the enzymes that modify histones. Kind of like having a librarian who doesn't know how to label books properly.
So if the writers and erasers aren't working right, it can mess up the whole system.
Exactly. But it's not just about genes either. Our environment and lifestyle can affect the histone code and DNA methylation patterns too.
Hold on, you're telling me things like what I eat and my stress levels can change how my genes are regulated.
You got it. There's more and more evidence that things like diet, exercise, exposure to toxins, even stress can change the epigenetic landscape. For example, studies have shown that eating lots of fruits and veggies can lead to good histone modifications.
So eating our veggies can literally help keep our histone code in good shape.
Seems like it. On the other hand, if you're stressed out all the time, that's been shown to cause DNA methylation patterns that make you more likely to get certain diseases. It's like stress is putting extra locks on that instruction manual so the cell can't get to the info it needs.
So that means we have some say in our epigenetic destiny. right? It's not just the genes we're born with, but also what we do and where we live.
That's a great way to think about it. And it shows why it's important to understand this stuff, not just to treat diseases, but to prevent them in the first place.
Okay, we've covered so much. Histones, chemical tags, writers, erasers, readers, chromatin modeling, histone variants, DNA methylation, and how all of that can be affected by our genes, environment, and lifestyle. It's pretty mind-blowing to think about how much is happening inside ourselves.
It's definitely a fascinating and quickly changing field, and the more we learn about these things, the more we realize how complex and well coordinated our biology is.
It really is. It's amazing how all these processes work together to keep everything running smoothly. But with all this talk about broken writers and erasers and locked up instructions, it makes me wonder, can we actually fix these epigenetic errors?
That's a really great question, and it's what a lot of researchers are trying to figure out right now. The idea of using the epigenome to treat diseases is super exciting.
So we could actually rewrite those messed up instructions and unlock important genes that have been shut down.
That's the hope. There are already drugs being developed that target these epigenetic mechanisms, especially for cancer.
So how do those drugs work? Are they like specialized librarians going in, fixing all the wrong labels?
Kind of. Some of them are designed to block the enzymes that remove those highly recommended stickers. Remember the HDAC inhibitors we talked about.
Right, so if you block the erasers, you're basically increasing the expression of genes that might have been silenced.
Exactly. That can be really helpful for cancers where the tumor suppressor genes have been epigenetically silenced. If you can turn those genes back on, the drugs can help slow down or even stop the tumor from growing.
That's amazing. So we're not just treating the symptoms, we're trying to fix the underlying cause.
Yep. And it's not just for cancer either. Researchers are looking into epigenetic therapies for all sorts of things: neurodegenerative diseases, autoimmune disorders, even mental health conditions.
Wow, the possibilities are endless. But I'm guessing there are some challenges too, right?
Definitely.
Hmm.
One of the biggest ones is being specific. The epigenome is so complex and everything's connected. We have to make sure that when we try to change something, we don't accidentally cause other problems.
So it's like trying to fine-tune one little thing on a giant control panel without messing anything else up.
Exactly. We need to really understand the specific epigenetic mechanisms involved in each disease and then carefully design therapies that can change those mechanisms in a precise way.
It sounds like there's a lot more work to do,
Yeah.
but the possibilities are incredible.
I agree. This field is changing so fast, and it has the potential to completely change medicine.
Well, this deep dive has been amazing. We started by thinking of DNA as a simple blueprint, but now we understand how it's packaged up and tagged and regulated in such a complex and dynamic way.
It really shows you the beauty and complexity of biology and how incredible science is for helping us uncover these secrets.
And I feel like we've only just scratched the surface. There's so much more to learn about the histone code and DNA methylation and all the other parts of this incredible epigenetic world.
Definitely. There are so many opportunities for new discoveries in this field, and I can't wait to see what we find out next.
It's inspiring to think about how new therapies could target the root causes of disease and really change people's health for the better.
It really highlights how, even though our genes are the blueprint, we have a lot of power to change how that blueprint is used. The choices we make, the environment we live in, even our mindset can shape our epigenome and ultimately affect our health and well-being.
That's a great point to end on. It's not just about our genes, it's about what we do and the choices we make.
Exactly. It shows that we're not just along for the ride, we're actively involved in shaping our own lives.
Well, on that note, I think it's time to wrap up our deep dive into chromatin and the histone code.
It's been great exploring this topic with you.
Thanks so much for being here. And to everyone listening, we hope you've enjoyed this journey into the amazing world inside ourselves. Until next time, keep exploring the wonders of science and the power of knowledge.